a product manager for the molecular biology tools at Benchling. And today, with my colleague Damian, we're going to be telling you about the molecular biology tool. And actually, Damian is going to be walking through a live example of using different aspects of the tool, from primer design to codon optimization to viewing and alignment as well. All right, cool. So just to set the stage a little bit for today, we're just going to be spending a little bit of time walking through some of the molecular bio features that we've released in the last few months. And then Damian will take over, walk through his live demo with all of you guys, and then we'll return back and talk about a few roadmap items for the rest of the year into the beginning of 2020. So I realize that this is now probably the second time that you guys have seen these slides, so I probably won't talk too much about this, but as you know, we were founded in 2012 and we've grown tremendously since then. And today we're going to be focusing mostly on the molecular biology set of tools, which, as you may or may not know, is actually a suite of a bunch of different tools, ranging from primer design, plasmid construct design, cloning tools, guide design for CRISPR, sequence visualization and alignment and validation. And just to kind of talk about a few new things that we've introduced, or new features we've introduced lately. We've highlighted all these guys in red here to show things that we've made updates to in the last few months. We'll be walking through our new codon optimization tool, and also showing you some new bulk assembly features we've integrated recently, and I think Ashoka has already mentioned work lists as well, so we'll briefly talk about that as well. So just to highlight two new features that we've just released, codon optimization in Benchling. Now you can codon optimize and back translate all of your sequences, and we currently have a bunch of different parameters that you can add in right now. For example, you can enforce or preserve certain restriction sites, and we've already identified a bunch of additional parameters that we'd want to enable in the coming months, so we can talk about those in the roadmap section. And as Ashoka already mentioned in the introduction of work lists into Benchling, work lists are a great way to combine a bunch of different entities from different origins, from different folders, and basically use that single work list as input for all sorts of different bulk actions. Today we're going to be talking mostly about bulk assembly with a work list full of sequences, and Damian will show you how by adding sequences to a work list, this basically speeds up the process of setting up a bulk assembly a lot. So this is the example walkthrough that Damian's going to be going through in a little bit. We're going to be optimizing and expressing vanillin in E. coli. So what this is really going to entail is codon optimizing vanillin dehydrogenase, creating a bunch of plasmids, annotating those plasmids, and preparing them for assembly, and then attaching primers, designing, and then finally sequencing and validating what we got. And in doing so, we'll walk through a bunch of different bulk and high throughput features we've added recently into Benchling. For example, work list integrations for bulk assembly, bulk auto annotation, bulk primer attachment, and then bulk alignments. So with that, I'll hand it over to Damian.
Hello everyone, so my name is Damian Costello. As Andrew mentioned, I'm a customer success representative with Benchling. Looking out, I see some familiar faces. I'm going to be walking everyone through this kind of very interactive live demo. For those of you who signed up for this track, you might have gotten an email yesterday prompting you to log into this BenchTalk 1 Benchling platform. If you've done that, we've created a folder for you with some sample data for you to play around with. If you did not receive that email or did not sign up in time, that's quite all right. You can follow along and kind of get some exposure to the molecular biology tools. Alternatively, if someone's willing to share their computer with you, you guys can tag along with me. Before I go too far into the workflow, I actually want to just take a step back and talk about some of these tools. So our molecular biology suite has just a wide variety of tools, right? Some of these, of course, we're not going to get to explore today, but I think what's critically important to understand is that we have a very engaged user group who provides us with a lot of feedback and a lot of constructive feedback to help us develop these tools, right? So as you're going through and you're seeing some of these feature enhancements or just a tool that exists in our molecular biology suite, I hope you understand that that is a product of the extensive feedback that we've gotten from our community. And so much of the growth and evolution of our molecular biology tools are because of the collaboration we have with our end users. So that's the first thing I wanted to kind of point out. The second thing I want to just address is that we really want to show you how you can kind of use high throughput sequencing design using our molecular biology tools. It's not just some disparate set of tools like a PCR tool, a primer design tool. You can actually kind of create these end-to-end workflows using our molecular biology suite. And of course, we want to showcase some of the new features as well. So without further ado, I'm just going to come back through here. What we're going to do is we're going to come in. I'm actually going to start and show you guys how to import sequences in bulk into Benchling. It won't require any action on your end. Then you guys can go ahead and sign into benchtalk1.benchling.com following the login credentials you set up the other day. You'll be able to actually find a folder with your name on it, which contains some curated data, including the vanilla dehydrogenase gene that we're going to codon optimize. We'll then use, as Andrew mentioned, the bulk assembly tool to essentially design these vectors, assemble them, do some annotation and primer design, and finally end with alignments. So what I'm actually going to do right now is I'm just going to jump into the platform. So if you haven't already, please take some time to come into this platform here. When you do so, you'll be able to click on this project side panel. And you'll see, you might not see these here, but you should see the Advanced Molecular Biology Workshop project. If you click into this project, what you might notice is a variety of different folders. So again, if your name isn't there, your folder isn't there, that's quite all right. You can just follow along visually. Alternatively, if you're able to find your project folder, you should see a list of sequences. One thing I actually have to do right from the beginning here is make sure everyone has the correct permissions here. So just like a little bit of a side piece here, you actually can set project permissions across any one of your projects to make sure that folks aren't editing your sequences and things like that, right? So you can set pretty granular permissions. So again, you should be able to find this here. While folks are kind of getting situated, I just want to point out, real quickly, one of the things that we're not going to be covering today is kind of bringing sequences into Benchling. Of course, all of the data is curated in Benchling for you already. I know, we've gotten a lot of feedback that given that Vector NTI is sunsetting in 2020, a lot of folks are moving away from Vector NTI, trying to get their, archive files out and into new sources. And so I thought, a relevant example of kind of bringing sequences into Benchling would be using this kind of Vector NTI archive file that I have. So real quickly, I'll just show you, you can imagine, coming from any, other external database with some sort of archive file. Within this archive file is a list of a number of sequences. What you can imagine doing is actually just simply coming to this inventory section. You'll see here that I've actually created my own subfolder here. I can come into here and you'll see, I have some nested subfolders, one of which is this Vector NTI backlog. I can actually just click into this and use our create button to import these sequences, right? So again, real quickly, just showing you, I can choose this import DNA sequence dropdown. Of course, you can import primers and amino acid sequences as well. And with just kind of this simple drag and drop, you'll see that I'm able to bring in a variety of different sequences kind of all at once here. We do support, a variety of different archive files, including mixed archive files, which can contain DNA and amino acid sequences. And just to kind of give you a quick visualization here, I can, open a particular sequence. You see that it carries over all of those annotations, any descriptors, anything associated with the sequence, right? So just wanted to kind of take a step back and show you that first piece, which is bringing sequences into Benchling. So for this next part, you guys who have folders here, please feel free to follow along. One thing that I do suggest is that you'll notice that there is this kind of display icon here. When I click on this here, it gives me these kind of display options. I highly recommend actually sorting the folders that we have here in alphabetical order. This will just kind of make the user flow a little bit more friendly as we continue on, and it also calls out that you can kind of organize folders and sequences in a fashion that makes sense to you. So I do recommend kind of putting these in alphabetical order if you haven't already. Okay, so now what you can do is you can click into your specific folder, and you should see this vanilla dehydrogenase gene sequence. So what you can actually do is click on this sequence to open it. You should be seeing something like this. I just want to call out too, one of the biggest entries of barrier with the Benchling platform can be just the way things are displayed, right? So as a way to give yourself a little bit more real estate when you're working with a particular sequence, you can actually just click on this project side panel here to collapse it, and you see that kind of opens up the space for you, right? So now you have a little more space to work. So as we previously discussed, the first step is going to be for us to codon optimize the vanilla dehydrogenase gene. So what we're actually going to do is click on the VDH annotation to highlight that entire sequence, right? The next thing that you're going to want to do is right-click on the sequence to kind of expose this drop-down that you're seeing here. You'll notice, if you haven't already, that at the bottom of this drop-down is an Optimize Codons option. So of course, this kind of being one of our first major feature enhancements in the last couple of months. You can click on Forward, and what you'll see is actually the side panel that appears on the either right or left-hand side of your window. It's this kind of codon optimization preview window, right? So as you can see, you can toggle different parameters. You might have a particular organism that you want to codon optimize for. In this case, we'll be codon optimizing for E. coli. You can set some sort of criteria for GC content. In this case here, I'm just going to keep it as any GC content. With respect to immunogenicity, you can choose to toggle mRNA uridine depletion if you want. And then at the bottom, you'll see kind of these three options for protected regions, avoided cut sites, and preserved cut sites, right? So protected regions, of course, dictating whether or not you want to conserve some region within the codon optimized sequence. Avoided cut sites is actually going to remove any particular restriction enzyme sites that you see in the sequence and prevent these restriction enzymes from being introduced in your optimized sequence. In this case here, we actually have 2 POW1 restriction sites. So if everyone can just click on this Add Cut Site to Avoid, you'll see this kind of restriction enzyme search box appears. We can just search for POW1, and we can avoid this particular cut site. The next step then would be to preview the optimization. What Benchling is going to do now is bring open this new optimization or updated optimization preview window. You'll see that we provide some metrics on the number of rare codons that you might have, GC content, the uridine content in the sequence before and after, as well as a variety of different codons that were manipulated in the original sequence compared to the optimized product. You can, of course, go back and update your parameters if you so choose. In this case, I might agree that this particular VDH gene looks good. I want to go ahead and save it, right? You can save this as a new sequence if you would like to. In this case, we actually just want to save the original sequence. So what I can do here is click on Save, and you'll notice that the sequence updates automatically. As an aside, there's actually something that I want to call out. So Benchling uses this version history to track every single edit that's ever made to your sequence. So no edit is ever lost. There's a running audit trail of who made what action to your sequence at any given time, and you can always revert to a previous sequence or clone a version of a sequence if you so choose. So just something I wanted to call out for anyone who is maybe in a legal or compliance team. So moving right along, the next step for us then would be to kind of bulk assemble this particular sequence into a variety of different backbones. So our bulk assembly tool is actually what we call like this combinatorial vector design tool. It allows you to combine a variety of different backbones and promoters and any combination of fragments that you can think of with just a couple simple clicks. There's been a variety of feature enhancements that we've made to the bulk assembly tool that I'll talk about in just a second. One of the ones you've heard about a couple of times now is this integration with work lists. So before we move forward, what I'm actually going to do is show you how you can actually add this particular codon optimized gene to a work list, right? So again, what I've done is I've clicked on this project panel. You can see that in Benchling, we do have this concept of a save state. The last subfolder that I was clicked into remains open in this project view. When I click on this caret in the upper right-hand corner, what that's going to do is bring open this expanded window. And this is actually a pretty important concept for us when we think about high-throughput design. This expanded window not only allows you to manage where those sequences are placed, where those notebook entries are placed, but also what sort of analysis or action you want to take against a particular sequence, right? So what I can do here, for example, is I can click on this VDH gene, and you'll see on the right-hand side I have a variety of options. I can move this to a new folder, I can copy it out to a different folder, I can archive it if I so choose. Alternatively, I can actually add this to a work list, right? So what I'm going to do here is I'm going to click on Add to Work List. You can see that I don't have any existing work lists, so what I'm going to do is actually create a new one. Please feel free to try this out yourself. I will put that this is my particular work list, and I'll add it, right? So you can imagine doing this for a number of different sequences spread across a variety of different project folders, etc., right? It's another way of thinking about it is kind of your favorites list. And now what I'm going to do is I'm going to navigate to the bulk assembly tool. So the way that you actually access our bulk assembly tool is by clicking on the Create button on the left-hand side, hovering over DNA sequence, and you'll see at the bottom you have an option for bulk assemble DNA. When I click on bulk assemble DNA, you'll see this kind of user interface appear. So one of the there's a variety of different features that we've built out for the bulk assembly tool, as I had mentioned, one of which is now this work list integration here. So you actually have the option to pick from particular folders the fragments that you want to assemble. Alternatively, you can add from a work list. You can see here that our steps are pretty much divided by the number of fragments that we want to insert into this vector. In this case, what we're going to be doing is using 2 common backbones in combination with 3 different promoters and our codon optimized vanilla dehydrogenase gene, right? Of course, the first step is to pick our backbones, and we wanted to show you how you can kind of pick fragments using both of these options here. So we'll begin by picking our backbones using a project folder. So what you guys can actually do is you'll notice that on this advanced molecular biology workshop, there is a little caret to the left-hand side of the project. When I click on that, what it actually does is provide me with a drop-down of all of the project folder or subfolders living within this project. And you'll notice that we have this folder called a bulk assembly folder. So please feel free to use this to grab the fragments that we're going to need for the bulk assembly tool. So what you can do is once more click on this caret, and you'll see that we have a variety of different fragments here, right? What you can do is go ahead and click on the low copy and high copy backbones. Next, what you can do is you can scroll down to kind of the fragment direction that you want, as well as the assembly method that you're going to do. In this case today, we're going to be doing a basic restriction enzyme-based method of assembly. We offer enzyme. This is great for classic RE-based cloning, as well as any Golden Gate assemblies you might do. Homology, very popular for Gibson assemblies. And then we offer this assembly method called concatenation. Concatenation does not represent any of the cloning techniques that you might use at the bench. It simply allows you to stitch fragments together independent of restriction enzyme overhangs or homology regions. Again, for today, what we're going to do is we're going to pick restriction enzyme based, right? The first restriction enzyme that you're going to want to choose is XMAJ1. Right? And the second one that you're going to want to choose is POW1. What you'll see is actually one of the other feature enhancements that Benchling has built out in the last couple of months. It's actually the display feature that you see on the right-hand side. This is showing you which of the fragments you intend on keeping, right? Again, if I choose this, reverse orientation, you'll see that the direction of the fragment changes. I can also choose to keep the larger or smaller piece, and Benchling will update that for you automatically, right? So this is actually one of the popular feature requests that we heard from users that we went and built out in the product. Okay, so again, we were picking XMAJ1 and POW1 here. The next step would be for us to pick our promoters. Again, you can just click on this insert tool. We'll just pick from a folder one final time, and then we'll move to the work list. So following the very same action, you'll click on this caret here. You can pick the bulk assembly folder here, and you will choose the particular pieces within it. One of the other kind of UI feature or enhancements that we built was the ability to actually click on a folder and highlight everything within that folder if you so choose. Alternatively, we now support shift select or shift clicking to highlight regions as well to make this kind of a little more friendly. In this case here, we're going to be picking these P-BLA, A, B, and C promoters as our fragments. Again, we'll be picking restriction enzyme, and you might notice that at the bottom of the enzyme text box is a currently used kind of feature where you can select restriction enzymes that you had used on the previous piece. Again, this is yet another kind of UI enhancement that we've built out for folks. If you're using homology-based assemblies, we'll remember the homology region of your sequence as well. Later on down the line, too, we'll kind of talk about roadmap items for homology assemblies in particular. One of the features that we hope to release soon is a homology detection tool that will recognize homology regions within your sequence and present it to you so that you can select those without having to paste the bases in. So moving quite along, we're here assembling our promoters into this particular backbone. We're going to pick XMAJ1 for our first one. Since this is a three-step assembly, the other enzyme that we're actually going to be using is BAMH1. You'll notice that again, these are, in this case, linear fragments. Benchling is showing me that they're flanked by these particular regions. They'll show me the left overhang, the right overhang, as well as the length of the fragment that I've selected. And you'll notice that Benchling will provide you with some sort of messaging on whether or not your assembly will work. So because we've picked BAMH1 in this case, our overhangs do not match, and this won't be resolved until I insert the last fragment here. So finally, we have the third insert here. What I'm going to do now is show you how to do this from a work list, right? So it's actually pretty straightforward. I really like this. As someone who's been using the bulk assembly tool for some time, this is pretty exciting. When you click on the work list tab here, what you'll be able to do is actually search through the work list that you've created. In this case here, I'm going to choose the work list that I just moved my vanilla dehydrogenase gene to. You'll see that that appears here. The last step for me then is to select my restriction enzymes. I'll pick BAMH1 and POW1 here. It'll highlight this region for me, and you'll notice that now all of these errors or error messages have been resolved. So in this case here, we took 2 common backbones, 3 promoters, and 1 common insert to make a total of 6 unique insert genes, or vectors rather. The last step here is for us to finalize this assembly, right? So you can choose a folder that you would like this particular sequences to live in. In my case, I want to save it to my particular folder. Please feel free to save it to your folder if you'd like to as well. I can choose select. And one of the kind of cool features that we built out that we won't dive into too much here is kind of an integration with the registry, right? So in this case, if I had a plasmid schema, like Tara had described in the previous seminar, I could set that plasmid schema to all of these vectors that I've just designed, and populate these sequences with pre-filled metadata fields that I could go then and capture critical metadata against all these vectors that I'm designing, right? So a really nice kind of registry integration that we've built out too. Finally, what I'm going to do is I'm going to click on Assemble. And what's critical to know about clicking on this Assemble button is that Benchling is going to open these assembled sequences in the expanded window. And again, this is because Benchling is prompting you to take some bulk action against the vectors that you've designed. If I click into a particular sequence, I can see that here is my backbone. I have this VDH gene. If I ever wanted to kind of automatically tag this sequence with other genetic elements or features without having to go through and find them manually, for example, I want to know where my promoter is in this backbone, I can use Benchling's auto annotation tool, and I can do this in bulk. So what I can do is, again, from the expanded window, shift select all of the vectors that I've just designed. I can come over to this More dropdown on the right-hand side, and I can choose Analyze. And this Analyze tool is going to be your best friend when you're doing any high-throughput design or analysis in the mobile suite, right? You'll see that we have this option here to auto-annotate. When I click on auto-annotate, you'll see that I have this feature library. Feature libraries are essentially a way for you to associate sequences with a particular tag, store that in a particular library, and have Benchling look for those sequences on your map and associate it with a tag. So in this case here, I can choose backbone features and auto-annotate. What Benchling will then do is auto-annotate all of these sequences. If I click back into a particular sequence, you'll see that I have my P-BLA promoter here, as well as some amp resistance and origin of replication annotations, right? So Benchling has gone through and done this work for you. It's found these sequences for each one of your plasmids and annotated them accordingly. The first thing to keep in mind is that if you're bringing in a sequence file from, let's just say you have a GenBank file or a FASTA file, any annotations that already exist on those sequences will actually come over once you import them, right? So in this case here, this annotation ribbon on the right-hand side will have a list of all of the annotations that were associated with this backbone when you brought it in from your external source. Another thing to keep in mind is actually if you're bulk assembling fragments that were pre-annotated, those annotations will keep, right? So if I would have done this with those annotations, you would have seen them automatically. I just kind of wanted to demo these here. What you can actually do is you can create a feature library. Essentially, what that would entail is just naming it. And you'll see that there's this expanded annotations window. I can actually take all of the particular annotations that I have, shift select, and save them to a particular feature library automatically. Alternatively, I can, find an annotation on my map here. I can right-click on it, add this to a feature library as well. The last method you have for kind of adding these annotations to a particular feature library is to import a CSV file that has just the name of the annotation that you have as well as the basis. A lot of our clients and academic users as well will export sequences and features that come in a CSV format, which is why we support it here in the platform. Creating new annotations is also pretty simple. If I wanted to do that, I could actually just highlight a particular region, right-click, and choose Create Annotation. I could say, and this it's not, but in this case I could say it's an open reading frame. I can save it. I can right-click on it and add it to a feature library, etc. Yeah.
How easy is it to delete that?
It's pretty easy. So I have a couple options. I can, again, use Shift Select here to delete, a particular subset of them. Alternatively, I can actually just right-click and choose Delete Annotation. Yep. Yeah.
You create a bulky files here, but the name is already in the tool automation tool, right?
Yes.
Do you know how easily increase the list of names?
The behavior for this now is just a concatenation of the fragments, as you can probably see here. Okay, So now we've annotated these sequences, we've gone through and just made sure we can associate particular regions of our vector with these tags. The next thing that you might want to do is, attach some primers that you've designed. This is really powerful because it's a collaborative tool, right? If you're working with other scientists, they've designed a primer for, let's say, a common backbone that has the U6 or T7 promoter. Somebody already has designed the primer. Instead of you taking the time to go and design your own, you can have Benchling check and see if that primer has already been designed and if you can attach it to this particular sequence. So what we can do is we can highlight, again, these particular sequences here. Very similar steps, right? Coming to the More dropdown and choosing Analyze. You'll see that we have this option to attach primers. For you guys, it might look something like Add Locations here. So go ahead and just select Advanced Molecular Biology Workshop. And what that's going to do is that's going to have Benchling look through every one of our subfolders that has a primer and see, hey, does it match, right? You can see that we have these, constraints that you can set. And finally, you can choose Attach Primers. So now, if I actually, click into one of my sequences, you'll see that these primers appear at the level of my sequence. If you are not seeing this, just a quick aside, make sure that the primers is toggled on under your display features. But you can see that Benchling has come through and attached primers where there was coverage for these particular sequences. Now, I did this in bulk. You can also do this for an individual sequence if you want to. Now, in this case, let's say, hey, great, we have coverage all over the place, but one area where we don't have coverage is the actual vanilla dehydrogenase gene, right? So maybe I want to design new primers in bulk to sequence this vanilla dehydrogenase gene. You can actually use Benchling's Primer Wizard to design primers all at once for this particular sequence. I just clicked on this VDH gene to highlight it. And I'm just going to close this to give myself a little more space. So again, I've clicked on this to highlight the VDH gene. I can come over to, kind of tough to see, but this bidirectional arrow on the right-hand side. You'll see the primers that I've already attached on the level of the sequence, and you'll see that I have an option to create additional primers, right? For today's demonstration, I'm going to show you the Primer Wizard. The Primer Wizard is really a tool that allows you to set really granular parameters around, how your primer should be designed. For example, I can pick sequencing primers here if I wanted to. I can tell Benchling to use a selection. What this is going to do is look back at the region that I'm highlighting and say, hey, in this entire area, design primers for me. You can click into this and set, minimum GC and melting temperature criteria, ideal, optimized GC content, melting temperature, size, right? You can tell Benchling to generate a certain number of results. I tend to leave this one alone simply because I want to set my criteria here and let Benchling decide, what primer would fit based off the other criteria I have. And then finally, you have this option if you're doing sequencing, like we are today, for spacing, interval, lead, and accuracy. All of these are actually defined by Primer 3. We have a manual to it here. But what these are is essentially telling, Benchling how far between you want primers to be designed. Spacing, for example, means create a primer every 300 base pairs or 750 base pairs from the 3' end of one primer to the 3' end of another primer on the same strand, right? So you can set really granular kind of parameters for how you want to do this. In either case, you can click Generate Primers once you're done. Benchling will look back and will provide you with a list of the particular primers that could be bound to this region that I've selected. Another feature enhancement that we've built out here is to allow people to, use specific forward and reverse primers if they so choose, rather than having to select the entire primer pair, right? So you can come through here, you can pick these particular ones, and you can save the selected primers, right? In this case here, I might save it and attach it to my particular folder. So what have we done? We've imported our sequences into Benchling, we've codon optimized them, we've added some to a work list, we've gone to the bulk assembly tool, assembled all of these vectors. We attached sequencing primers there and designed new ones to cover, regions that didn't have coverage. You can imagine going to the bench, kind of prepping these samples, mini prep samples, for example, to sequencing. And then you might, submit to a CRO or to a sequencing core, or you are the sequencing core, and you want to now visualize the alignments to confirm that you've made at the bench what you intended to make. So the last step then is to just show off the alignment tool. So what you can actually do here is go ahead in each one of your particular folders. I'm just going to go ahead and, for example, click on Erica's here. You'll see that you have these templates as well as these CRO AB1 files. So what you can actually do here is click on the expand arrow one last time, select the following sequences, come back to the oh so familiar more dropdown, analyze, and we're going to create a DNA alignment. What that's going to do is that's going to bring all of those sequences that you've just selected into a window here. You can import additional sequences from your computer. You can search for a particular sequence that already lives in Benchling. You can add additional sequences here by manually or by copy and paste. And then finally, you'll see that Benchling by default wants to create a consensus alignment. This is because we've done this through the expanded window. Of course, consensus alignment is going to create a unique DNA sequence in Benchling. In this case here, we want to align to a template. So to do so, I can just simply click on template to set it as my template. It's very easy to toggle on and off. Once I've done that, I can pick a particular, alignment algorithm that I would prefer to use, for example, MAFT or Costello Omega in this case. And finally, I'll choose, create DNA alignment. What that's going to do is open up something like this. And again, I'm just going to close this. I want to take, just another second to talk about the alignment tool. We're not going to have time to go through all of the feature enhancements that we've built out for the alignment tool, but this is by far one of our most popular tools. And we've done, quite a lot to make this a more friendly tool to work with. So you'll see that down here we have this kind of expanded mini map showing me my contigs. Anything that appears here in red represents a mutation. Anything, not in this case, but anything that appears in yellow would be an ambiguous base pair. One of the features that we're shipping very soon is the ability to jump to mismatch. So in this upper right-hand corner here would be an arrow to automatically jump across all of the mismatches in your alignment. You'll notice that I can kind of click and drag this if I want to. I can see, particular regions that have mismatches. I can make base calls directly to a particular AB1 file if I want to here. In this case, I just change it to the same thing that it is because I'm not thinking straight. So I can change this here if I want to, right? You can make base calls. You can see that annotations are automatically displayed here. A lot of the display features you see at the level of the sequence can be shown here as well. So you have a variety of different options, right? Each one of these sequences, too, can have its own particular, display feature shown. So that's, kind of a unique feature, too. We also have a new feature that you can actually right-click into a particular region. You'll see that you can trim. You can set your trim to the start and end of all of the sequences or to the start and end of a particular sequence. That will drag this trim out. So you have a number of different, options here for kind of toggling display and alignments. All of these alignments are then saved to the template map and can be referred to at a later time. Yes?
Why is the files of the consensus sequence?
That's a great, so if it's a consensus sequence, what it's going to do is create a unique DNA sequence like this. If you're aligning to a template, in this case here, I didn't import them using AB1 files, but the AB1 files will be attached in the description tab of the template map. So you'll be able to download them back out. Alternatively, I actually imported these AB1 files into a particular project. So if I wanted to, I could actually, come back to the expanded window and export all of these sequences back out if I need to. Okay, so I'm going to hand it back over. We have about 7 minutes left for kind of Andrew to go through some roadmap items. We do have a little bit of time after this, so if you guys want to meet and talk about specific use cases, things you really like, feedback that you have, happy to do it. thank you very much for kind of giving me an hour-ish of your time to kind of walk through some of these features we're excited about. And I'll hand it back over to Andrew.